Today I'm going to tell you about our study, Variants at the OCA2 HERC2 locus affect time to first cutaneous squamous cell carcinoma in solid organ transplant recipients, collected using two different study designs. This was published in British Journal of Dermatology and was a multidisciplinary and collaborative study. Our study team comes from three institutions: The Ohio State University, University of California, San Francisco, and the University of Minnesota. Cutaneous squamous cell carcinoma has long been recognized to be a significant clinical issue for solid organ transplant recipients. Solid organ transplant recipients have a 60 to 200-fold increased risk of SCC, increased mortality to cSCC, and increased risk of multiple skin cancers. Our study is looking at OCA2. Why study OCA2? It maps to a region of the genome important in skin cancer risk in mice. Variants in and near OCA2 are associated with increased risk of the three major types of skin cancer: basal cell carcinoma, melanoma, and squamous cell carcinoma in both candidate gene and genome-wide association studies. Furthermore, variants in OCA2 have been associated with blue eye color, skin pigment, and hair color, and pigment has a known role in protection from UV-induced skin cancers. Our research group is very interested in identifying genetic risk factors for SCC in transplant patients. We're specifically interested in answering the question, are OCA2, HERC2 blue eye color associated genetic variants associated with time to first SCC post-transplant? We are interested in looking at two variants, RS916977 and RS129-13832, as these variants have been associated specifically with eye color, skin tone, and SCC risk in previous genome-wide association and candidate gene studies. To answer our research question, we used data and samples from two studies: an OSU-based study in which we looked at 125 organ transplant recipients ascertained through OSU clinics, 67 with at least one SCC, and 59 with no SCCs, with an average of 12.1 years follow-up. The UCSF study consisted of 263 organ transplant recipients ascertained across the United States. 192 had a diagnosis of SCC, 71 had no SCC with an average of 9.5 years follow-up. We genotyped normal genomic DNA from these individuals for the two variants at the OCA2 HERC2 gene region that had been previously associated with eye color and other skin pigment phenotypes. Cox regression models were used to assess genotypes with time to first SCC and eye color as variables. We found that the blue eye associated alleles of RS129-13832 and RS916977 were associated with decreased time to first SCC post-transplant in solid organ transplant recipients, and this was significant for both variants. We also found, not unexpectedly, that both of these variants were associated with blue eye color in our study populations. But blue eye color itself was not significantly associated with either time to first skin cancer post-transplant or with overall skin cancer risk. This study is significant for a number of reasons. First, we found that blue eye-associated variants at the OCA2 HERC2 locus are associated with decreased time to first SCC post-transplant in two OTR studies, even though there were significant differences between the cohorts in terms of collection and study design. Second, this study has potential implications for the development of screening recommendations for SCCs and OTRs, particularly for timing of screening. Third, we still need to develop better tools to predict which solid organ transplant recipients are at the highest risk of developing SCC and when they are at increased risk. OCA2 HERC2 variants may add to the current models as they are more predictive for SCC timing than eye color.